And that's the best way to start, obviously muted. So hi everyone and welcome to this live stream. Today we will be talking about the tumor microenvironment. And before we start, say a couple of words about us, about Lab4Pose. Lab4Pose is a life science company and we are on a mission to help develop new therapies and diagnostics, especially for companion animals, and to reduce the overall use of animals in research, especially in human research. And we do this by doing our little piece, or giving our little piece of the puzzle, which is providing biospecimen, especially from companion animals, but also from other animals. And so what we provide are dissociated tumor cells from cats and dogs, for example, but also from other animals, healthy cells, especially used for in safety studies, also for in the development of human drugs, for example, when making comparison studies between human and animals to choose the right model. Frozen tissue, formalin fixed, paraffin embedded tissues, blood and then blood derived cells, biofluids. And then we have a portfolio of antibodies, ELISA, cytokines, all related to animal health and animal research. And then we also support Altis Biosystems with their human gut model, which is obviously a really important building block for organ chips, for example, organ chip system to reproduce the human gut in the lab, and helps also sparing some of the some of the animal testing. So let's get into the main topic of today, which is obviously tumor microenvironment. And when we are talking about tumor microenvironment, there will be like really a ton of different things to say, because the tumor microenvironment is not one single thing. It's a lot of different things together. And when we are developing new drugs, new solutions, or we want to understand more about a disease, then nowadays it's not possible anymore to consider, especially if we're looking at tumors, it's not possible to consider the tumor cells specifically individually. Once it was easier to just go out with a cell line and study that and really go in detail. And this can be obviously something useful in the beginning when doing part of the screenings. But if you're looking now at the new versions of the hallmarks of cancers, quite famous publication, basically mentioning all the different hallmarks of cancer, how we can recognize it. We see that there's quite a big change from the beginning, from the previous, or the first edition. And I think in the first edition, the first publication, there were 6 different hallmarks of cancer. And now altogether, if we put together the published hallmarks plus new, let's say, phenomena or other characteristics that have been recognized, we are up to 14. And funny enough, 5 of those at least are not really specific of the cancer cells themselves, but they're really dependent from the environment. And here, obviously, saying microenvironment, so basically the cells that are immediately surrounding our tumor, that would be the easiest thing to do. The easiest thing to say, okay, we have the tumor and we have the non-tumor just outside of it. But the reality is that the tumor itself creates a whole ecosystem, a whole system to maintain itself, which is much more than just the micro environment. In fact, it could, it becomes something a bit more systemic before it really becomes systemic in terms of diffuse tumor or metastasis or whatever. So what we can do today is to have a look at the different hallmarks of cancer and how these are important. So if we look at the initial publication, there were 6 different hallmarks. And the first one was sustaining proliferative signaling, which basically means that the cancer cells, they generate their own growth signals, they reduce dependency on external stimuli, and this obviously helps them. The second one is evading growth suppression. And they do this in a variety of different ways, but normally there are a lot of different mechanisms that are bypassed by tumor cells, and they get really good at it. The first one, if anyone ever used cells in the lab, they know it. In cell culture, they know that contact inhibition, cell contact inhibition is one of the things where cells at a certain point, they touch each other, they understand, okay, we are a little bit too tight here, and they just stop growing. For tumor cells, this doesn't really happen this way. Then we have resistant cell death. So this is in addition to the previous mechanism, where they basically do not just stop, they stop reacting to growth suppression mechanisms, but they also really avoid apoptosis signaling. And so they survive much longer than the normal cells. And one of the ways to do this, and this is the fourth hallmark of cancer, is enabling replicative immortality, which is what they achieve by stabilizing the telomeres. So they basically can continue replicating because they are, the chromosome do not shorten, the telomeres will not shorten over time. And then we have a couple of other topics already in the first 6 that are actually more related to the environment. And the fifth hallmark of cancer is inducing angiogenesis. So obviously, to grow in size, also tumor cells, they need the nutrients. So they need to be vascularized. Like the tumors, they need to be vascularized, and like blood vessels to get inside a tumor. And so this is a really important mechanism. And on top of this, there's the activation of invasion and metastasis. So cancer cells will acquire the ability to invade other tissue. To do this, obviously, they have to produce proteases, remodel the tumor microenvironment, and spread also in other bodies. So these kind of mechanisms, like epithelial to mesenchymal transition, these are really important kind of mechanism also for metastatization and tracking also this kind of proteins that are produced. It can be one of the most important biomarkers to track basically if the cells are going to migrate, what is going on with the extracellular matrix itself around the tumor. Then we have some new hallmarks that were published in 2011, and we have the reprogramming of the energy metabolism. And this is obviously relatively intuitive, if you want to put it like this, because tumor cells do not grow at the same speed as the other cells. They grow quicker, and to do this, they need a lot of energy. So they need to reprogram their energy metabolism to be able to grow that fast. And then avoiding immune destruction is also really important. So the development of mechanisms like the PD-1 ligand to basically contrast the activity of immune cells and even induce apoptosis in the immune cells. That's a really genius move, if you want to put it like this. That is really one of the things that is being tackled at the moment. But this is just one of the ways that the tumor interact with tumor cells, with the immune cells. And one of the other ways how tumor cells interact with their environment and microenvironment, not just at the level of the matrix of the cells that are there, but also at the cellular level, is by inducing a tumor promoting inflammation, where they basically attract cells of the chronic immune inflammation that will produce pro-inflammatory signals that will continue stimulating the growth of the tumor itself. And then obviously we have the genome instability and mutation. So we know that tumor cells would have a higher mutation rate compared to other cells. And they use this, obviously. They use this to evade immune mechanism. They use this to adapt. They use this to repurpose some of the mechanism of some of the proteins in another way, so that is more congenial, is more useful to them. And then there's a couple of others, of other hallmarks that are really important. And there are becoming more, some new concepts that are becoming more clear now. For example, the phenotypic plasticity of tumor cells. So is once they get to a certain stage, then tumor cells, they are much more they're much more plastic. They can change their phenotype better or differently comparing to other cells and adapt in other ways. And this is also complementary to the fact that tumor itself is quite variable because of this genome instability. Different cells inside the tumor are actually quite diverse. It's not always a single clone with all the cells the same, but there's also the idea that different cells inside the tumor might have a slightly different function. So this heterogeneity is also what makes, for example, dissociated tumor cells more interesting for some application comparing to a cell line, even if it's if that's a primary tumor isolated or like derived from a primary tumor, because it gives you more information at a single level of what is the role of the single cell, what is the transcription profile, the proteomics profile, and so on and so forth. Then we have the non-mutational epigenetic reprogramming, and this is something that is used by many cells, but it doesn't require to have like a mutation in the genome to really have an effect. Sometimes it's just enough to change the epigenome to basically reshape how the particular genes are expressed or not. And then a couple of other points that are becoming particularly important are the role of the microbiome, polymorphic microbiome. So a lot of different bacteria that are normally coexisting with ourselves might have a role into inducing a particular tumor type. And I think the most famous example is Helicobacter pylori with the gastric cancer, but there are probably a myriad of other cancer types that are created by or induced by the microbiome. And if not microbiome, there are also viruses like the papillomavirus virus that are inducing types of cancer. So this kind of crosstalk between bacteria, viruses, pathogens, and the human body, this is one of the most interesting also areas of research. And also, so when we are studying the tumor microenvironment, the tumor environment in large, then we want also to understand if that is an environment that is in a way conductive, in a way Okay, I think we are live again. That's the nice part of the live stream. You always have something unexpected. Exactly. So the last point is the presence of senescent cells. And senescent cells are obviously aging cells. And in fact, that they are aging, they are also producing factors in the microenvironment, growth factors and cytokines that are influencing that are having an effect on the growth of the tumor itself. And so this is about it today, about the tumor microenvironment. I will say a little bit more what are the different factors that we need to consider when we're looking at the tumor microenvironment. So it's not just about what are the cells that are in the immediate surrounding, but it's also about which kind of cell types, what is their stage, if they're young, are they old cells, are they human cells, are they bacteria, is there an effect of vascularization maybe? And vascularization means also oxygen concentration, CO2 concentration, pH, and a lot of different factors. And these are all things, obviously, that we're able to track up to a certain point, depending on how fresh is the tissue that we use, how was it treated, how was it preserved between the moment of the surgery and the preparation, and obviously the kind of work. So if I snap freeze the tissue, if I embed it in paraffin, it will have different kind of effects. If you're interested to know more, if you want to study the tumor microenvironment and you want to have any kind of comparison between tumor and the microenvironment or the normal adhesion tissue, then make sure to contact us. You will find our contact in, I think, the stream below. If not, it's info@lab4post.com, and we'll be happy to some support into the short Q&A. And we wait a second if there are questions coming. Perfect. Then having said that, I see that there are a couple of questions coming. We're running out of time, so we will answer by email. Thank you very much. And having said that, I wish you everyone a really nice evening. Talk to you soon and see you in the next stream.